that sometimes they're called soil transmitted helmets. We talked a little bit about the different kinds. I kind of showed you a picture of what they look like. So what I think would be good is for you to start thinking about identification. So if you see a picture, what it would look like, and I'll show you the pictures. I'll show you what the different types of helmets, what classes they belong in, and what they, how to identify them. Okay, so we're going to talk about flatworms and roundworms. So there's two different types of flatworms, and they are called cestodes and trematodes. So yeah, we can call them flatworms, but we don't really call them that anymore. These two belong together into the flatworm category, but they're called cestodes and trematodes. So cestodes look very different than trematodes, so they're very, very easy to identify. They're very simple to identify because they look very different. So I'll show you pictures, and the pictures will help you more than my drawings, but cestodes, just write the whole thing out. Trematodes look a little bit funky. They're like, sometimes they look like this. They look like little fish sometimes. So these are both flatworms, and they are flat. So tapeworms, even though it's called a worm, it is a worm, but they're like ribbon flat. They're very, very thin. And then we're going to talk about roundworms that are fatter, that are the traditional worm shape, that are nematodes, and that's it. But we have different kinds of nematodes, so we're going to go over the different kinds. Okay. So eggs, those are called ova. So where have we heard that word ova that we just talked about on your, does everybody have their quiz back? Okay, quiz is back. Where did we hear about that word? Yes, from the ova and parasite stool sample. Ova is the word for egg. So they have an egg, and they have a new type of life cycle called a larvae. So we haven't had this before. We only had cysts and trophozoites and protozoa. Now we have helminths that are eggs, larvae, and an adult phase, and it's called an adult. So they're not called like something special like trophozoite. Okay, so they are multicellular. They have organs and organ systems. So I'll show you what some of those look like. So if this helps you to put, however you put them into categories, whatever helps you to memorize the names and kind of what they look like. One more thing I wanted to bring up is the term helminthiasis. That's the disease caused by helminths, so it's called helminthiasis. Okay, so let's talk about the life cycle. So we have eggs or ova. Now as we move into helminths, this is one of their life cycles. We worry about this life cycle a lot because some helminths can lay millions of eggs, like hundreds of thousands of eggs in one helminth. So we worry about eggs a lot. Yeah, go ahead.
I was just going to ask on the slide before, the disease, the helminthiasis, whatever it was, I'm sorry. I can't.
Helminthiasis?
Is that for all different kinds of worms?
Yes.
It doesn't matter what the worm is, it's all the same name?
Yes, and that's a good clarification question, is that all parasitic worms that are parasitic, are called helminths.
Right, but I mean the disease caused by the different ones is still called the same thing?
Helminthiasis, it's always called helminthiasis.
Okay.
Yes, great question. Okay. I lost my little threadworms. I did threadworms, we're not doing threadworms. They look too much like pinworms. They're too confusing, so the Ascaris, pinworms, whipworms, and hookworms look very different. So you'll be able to tell the difference between those ones. Threadworms look a lot like pinworms, so it was too hard to tell them apart, and they're similar, and threadworms are less common, so yeah.
So they're like huge.
The huge ones are usually the cestos, the tapeworms. Those are the huge ones. Yes, those are the ones that are like feet and feet and feet and feet long and can live in the intestines very, very long. Yes, the Ascaris, they're pretty big too, but I'll show you they don't get as big. The tapeworms can get like from here to the stairs long in somebody's intestines. If not longer than that, here to the stairs that you walk up. Yeah, they can get extremely long, and I'll tell you why. Okay, so here's the eggs or ova. These are also a chitin-like material, so they also have this chitin-like material on the outside, similar to the cysts of protozoa. They can live 1 to 2 months outside of their host. They can live years in feces if they have some sort of nutrient absorption methodology that they can actually, because they're not fully dormant, they can actually survive, but they have to have nutrients as well. And they can't be killed with traditional wastewater temperatures. I just thought that was interesting. I won't ask you about that, but so a lot of times people will filter, so the wastewater treatment plant will filter out for larger eggs, for helminth eggs. Yeah.
What would those temperatures be like? Does cooking something kill it?
That is a good question. So I don't think wastewater temperature, I don't think they boil. I think it's less than boil.
Okay.
But let's Google that. If helminth eggs can be killed with boiling temperatures, I think they can.
Like if you stick something in the oven for at 350 degrees, is it going to kill the?
Yes. You know, yeah. Well, yes and no. Helminths are contracted a lot of times through meat, and if meat is cooked all the way through well enough to the given temperature that they suggest that you cook meat at, that will kill helminth eggs. That's a very good point. But let's look that up. The temperature that
it does.
Yeah,
it does.
It does. Do you know? Will you Google that? The temperature for me, really quick.
It was like it differs between different eggs. It says helminth eggs can be inactivated at temperatures above 50 degrees Celsius. But then
Ascaris, yeah.
Ascaris eggs can be completely inactivated at 70 degrees Celsius, 75 or 80 degrees Celsius.
Okay, so what is that? 75, 80 degrees Celsius?
100 to 212.
Yeah, so boiling is 100, so it has to be above boiling.
It just says boiling water, 100 to 212 degrees.
Okay. Oh, it says boiling water 102.
Yeah.
Oh, okay. So 100 would be the lower limit, right?
Yeah, this is saying 80 degrees Celsius to
Okay.
inactivate them.
Okay. So here's an Ascaris. That's what they're talking about. Ascaris is the roundworm. Okay. Which are also called nematodes. Okay, so here are the eggs. This is why we do an ova and parasite test. We look for ova and we look for parasites, which are protozoa. Okay, so now we have this new life cycle called a larvae. And we worry about this life cycle a lot. Well, and I just wanted to give you a little heads up also that it's not always these 3 definitive life cycles. Every helminth is a little bit different in their development and in their life cycle phase, but we're gonna remember these 3 because it's most. So most have an egg, a larvae, an adult, but some will have this intermediary weird thing that they call some unique name depending on what the helminth is. So larvae are unique in that it's almost like, so protozoa, when a protozoa is going to be transmitted from person to person or animal to person, they have to go through the encystment process, right? They have to get to form a cyst to be able to survive out in feces. Well, helminths are unique in that they need another step, and they need another step because they'll live in a different area, and then they want to get to their host as an adult. So they might need more than one methodology, more than one life cycle, so that they can survive in the adult and get into where they want to get into. So the larvae have different techniques of doing that, and the eggs have different techniques of doing that. So I'll just give you one example. So, like for example, sometimes people who are swimming will get eggs in their mouth and in their intestines, but if the eggs pass all the way through the intestines, then the egg won't survive and hatch. So the egg will just pass right through. But what the larvae does is if someone will swallow a larvae, or if the larvae is able to hatch, then that larvae will be able to live like in the stomach versus the adult will live in the large intestine. So they need like a second phase because they are more sensitive. Like we're moving up in sensitivity to where the areas that they need to be able to survive. So now they have this other phase that has to live in kind of an intermediate environment between where the host is happy, where the adult is happy in the host. But we worry about these a lot because larvae can infect different tissues than the adult, obviously. So we'll talk about burrowing and why that's important and where that comes into play. And then there's an adult phase. So the adult phase typically resides within the definitive host, the host where it wants to live. Then it will form the adult and it will go about its life of being happy in that area. So then the eggs can be secreted and expelled through the feces if it is living in the intestines. But also because of burrowing that we'll talk more and more about, you don't have to stress about it right now, but as we get later on the slides, you'll understand what I mean. So they can form these things called tissue granulomas. So it's just like a little tumor. So it's gonna form a tumor, but it's full of worm. So that worm is forming a little tumor underneath the skin, and that is where it's residing. So depending on where the adult is the most happy is where they will become an adult from their larval stage. So this is an example of a flatworm. So this is right here, the cestodes, and they can get feet and feet and feet long. And then here, I'm not sure what type of worm this is, but it formed a little tumor and it was living in this person's subcutaneous tissue. This is an intestinal ascaris, and we'll kind of go through all the different ones of those. So internal morphology of the helminth, now we've moved up to, this is our first time ever talking about a multicellular organism. So now these organisms, even at the larval stage, have some variation of the organ systems that we have. I put up there human 11 organ systems, which it's not our job to memorize that in this class, that's for your next class, or for AMP whenever you decide to take that. But they can have any variation on that. So they do have a rudimentary digestive system. They do have sometimes a rudimentary reproductive organs. Some of them have reproductive or a rudimentary nervous system. So depending on the organism, that they can have organs and organ systems similar to us. So they are multicellular. One little cell, like an epithelial cell, can't go off and live on its own. There's millions of cells that make up this organism. Yeah.
Do you want us to be able to recognize the anatomy or?
Nothing. Mm-hmm. No. Yeah, just know they're multicellular. They have organ systems. That's all I wanted you to know. And then just a couple funky things that I wanted to bring up too that I don't care that you know about either, but I just wanted to throw it out there, is that they have unique structures as well. So some helminths will have unique structures like an attachment organ or some sort of specialized reproductive organs depending on their environment and stuff like that. So Some have a full mouth and anus digestive tract, some don't. Some have organs of attachment that they can hook into the tissue, or they have suckers, some don't. Some have female and male parts in the same helminth and some don't. So just so you know, some have specialized structures that make them unique. Okay, so let's talk about them each individually. So here are our cestodes. So this is a tapeworm, and if you can see like the side view of how flat they are, these are flatworms. These are called cestodes. This is also, in layman's terms, it's called a tapeworm. There are different types of tapeworms. Here's the genus and the species of some of these cestodes. I don't want you to know any of those, but just so you know, there are different genus and species. There's a lot of different types of tapeworms, and these are depending on their tropic tissue of choice. They might be more likely to reside in pork, in pigs, or more likely to reside in fish or beef or things like that. So I'll tell you what we're looking for when I talk about organisms that exist in meat, because this is a new methodology that the organisms are utilizing that we haven't talked about yet, which is burrowing. Okay, so they're segmented, so they have these little segments. They're flat, they have a sucker attachment. These are called proglottids. So each of these little segments are called proglottids. And each of those little segments is actually like its own little living organism. So it's almost like super weird, right? But it's almost like a fungal hyphae, that the fungal hyphae can fragment and go off and produce its own fungal organism. Well, these proglottids are similar in that way. Like one proglottid is almost like its own individual organism. And then it will grow more proglottids, more proglottids, more proglottids. And then how dangerous is that, right? Because not only are there eggs in the intestines and in the feces, but also if a proglottid can go and reproduce and form its own proglottids, then it's just another form of transmission. Sorry, did you have a question, Dr. Myers?
I was just wondering if that was like to protect the organism, like if it got broken, it would just regrow.
Yeah. Yes, yeah, it can split off anywhere. Yeah, that's a good point. Yeah.
And the proglottid is what carries eggs?
Each individual proglottid is like its own little organism. So it carries all of its intestines.
Oh, I see. Okay.
Yeah, it carries like its intestines, its reproductive organs, its all that kind of stuff.
So a proglottid is in one segment of the segmented.
Yes, and a proglottid is fully capable of living and existing on its own. Yeah.
Are they connected while they're connected to the tapeworm to some kind of central nervous system?
I don't really know very much about the central nervous system, but I know that they do have like minor, like that they have like taxis, like they have phototaxis and chemotaxis, and they're able to have some sort of sensory perception, but that's as far as I know.
I guess I was more just curious, like when, because they're all their own organism, if they're all connected, they're moving as one somehow, so I imagine there's some kind of...
I don't think that there is, between the different proglottids, I don't know enough about it, really, to say.
Weird.
Yeah, it is weird, but I do, I don't know, I would just be guessing. Yeah, sorry.
So these can do asexual and sexual reproduction?
Yes, so asexual and sexual reproduction, all helminths can sexually reproduce in some way also, but they can mostly asexually reproduce. Yes. Yeah, and we'll talk more about reproduction next time too. Good question. Okay. So we don't talk a ton about cytopathic effects because what they're doing is they're existing like a true parasite. They're just living the dream. They're in there and eating your food. So some of them, when they're burrowing into the tissues, can secrete some proteases and things like that to be able to absorb the nutrients, but mostly what they're doing is they're eating your food. So a tapeworm, for example, if somebody has a tapeworm, they'll eat and eat and eat and can't gain weight because that tapeworm is just getting bigger and bigger and bigger, eating the food that that person eats. So it's a little bit different when you think about that. Okay, so here's their life cycle. They have adult larvae. They have this other stage that are called cysts, but I don't want you to remember that. We're just gonna only remember ova, which are the eggs, larvae, and adult. But some have these other forms also. Yeah?
Okay, I don't think we know this, but I'm just curious.
Yeah.
Since they're eating our food, and you said like they can't gain weight, will that person continually be hungry, or after they eat, would they be full and then not get nutrition?
You know, that's a good question because they don't live in the stomach, they live in the intestine, so they would probably be full, but then they just don't get all of the nutrients and they don't absorb all the fats and things like that from what they're eating.
So it'd be hard for them to eat more on top of that.
Yeah, so yes, and I do think they get a sensation of fullness also as the helminth grows because of the pushing on the organs. Yeah, but yeah, I don't know anybody that's had one. Yeah.
What's the difference between a helminth, cyst, and egg?
So we don't need to know anything about cysts, just forget about that. But just so you know, some have this other little life cycle. Yeah, just eggs, larvae, adult. Yeah.
What does larvae look like? Are they like?
So they look different in every organism, but this is called the scolex, which I don't care that you know, but if this little larvae stage starts growing these proglottids, and the proglottids can grow and grow and grow, and then if this proglottid will go out and exist on its own, it'll grow spout another like larval form, and then start growing its own proglottids. So it's almost like.
Is the larva like the head of it?
They don't really say head, but yes.
It kind of looks like that
way. Yeah, yeah.
Is that
And they're all a little bit different.
Is that what develops kind of into like the sucker maybe or something?
Yes, yeah.
Okay.
The attachment where it's got hooks and then it has suckers.
Okay.
And if anybody feels like vomiting yet, I'm with you. Okay. So intestinal, if they have been exposed to the intestines, what transmission route is that? Fecal oral. So they are fecal orally transmitted, yes, but also we have a new type of transmission that we're going to call foodborne. So even though one of our what are our seven fecal oral transmission routes?
Fomite, fluid,
Fomite,
feces,
food, fields, flies,
fluid,
feces was one of them,
yeah,
fluid, is that seven?
water.
And what?
The seventh, that's it.
Oh yeah, that's it, okay. Okay, so here, even though we have food, this is not the same. Because this just means there's feces on the food because of runoff or because of water that had feces on it getting on the food. Or maybe it was on the fly's feet and then the fly landed on the food. So it's not fecal oral transmission, it's not a part of fecal oral transmission, it's a whole nother beast. And this whole nother beast happens because of what we're gonna talk about is burrowing. So these organisms are able to dig into tissues, so such as the muscle of a pig, and then when that's rendered, which is the word for processing meat, and then someone eats that, and if it has the helminth in it or helminth eggs or larvae, then that's a transmission route. So it's different than fecal oral transmission. And then sometimes there's an intermediate vector. So I'm going to talk a tiny bit about intermediate vectors. So they might not have just one method of transmission. They might have to have two methods of transmission or else it will die. So sometimes they have to go through another phase, or the larvae will have to develop in another area, or the egg will have to develop in another area. So sometimes there is an intermediate vector, such as shellfish, or insects, pests, birds, things like that. So I'll talk a little bit more about intermediate vectors when we talk about one of the organisms is our next one, that it has to go through the intermediate vector or it will die. But this is also, these are also ways to transmit these cestodes, but our two that we're going to focus on are fecal oral and foodborne. Okay, so these are trematodes, these are flukes. These are weird looking. So these are they're like leeches. So they're not, even though leeches can get on the skin and that is a method of infection, some of them can go through the intestinal system as well and live in the intestines. And then there are different types of trematodes, and depending on their area of the body that they like to infect, it depends on the type of organism. So schistosoma is a more common one. I'll show you a picture of it. But these are also called flukes. So unfortunately, they have two names, flukes or trematodes. That's what they're called. So they can look like these little fishy things, or they can just look like a big flat thing. I'll show you another picture of what those look like. If it's flat, it's a flatworm. If it's round, it's a roundworm. Okay, so here's trematodes. They're non-segmented. They're flat. Sometimes it looks like maybe this would be a roundworm, except it looks like all the air's been like, oh, and it's flat. So sometimes they look like that, or sometimes they look like this, or sometimes they look like leeches, but they're all the way flat. So these ones have to have multiple hosts. So typically snails, so this is a big one for this one, is that they have to spend a part of their life cycle within a snail. So why do we care about that? One, snails are everywhere. So if a snail is an intermediate vector, if the snail dies, or if the snails crawling through the garden, then it can infect the whatever, the snail dies, for example, and it's in the soil, then a person can get that, or an animal can get that. But also people, which is more rare, but people who eat snails, they can get the transmission route that way. Okay. Some are oncogenic. Some can actually lead to cancers if they live in a human for a long time. I don't care that you know that, just interesting. Sexual and asexual reproduction. Most are hermaphroditic. I don't care that you know that either. They have a rudimentary digestive tract, blah, blah, blah. So the big thing about this one is the snail, one, because that's a transmission route for people who eat that, but also because snails are everywhere, and that's a very common method of transmission for flukes or trematodes. So the larvae can be free swimming, which I'll bring up what the problem is with that as well. So the larvae has its own tropism separate than the adult, and this is more common actually. The larvae sometimes can be more infectious than the adult. The larvae sometimes can burrow more frequently or live. They have like just like worms do. You unearth an earthworm, it's going to automatically crawl down back into the soil. It's just its nervous system. That's what its nervous system does, is crawl back in the soil. Well, same for flukes, is when they're unearthed in the tissue, then they want to dig back into the tissue. So especially for the larvae. So transmission, fecal oral. So remember, we're looking for ova. When we're talking about fecal oral transmission, we're looking for the ova, in ova and parasite. The reason I keep bringing that word up is because the ova and parasite stool sample, test is how we're going to find these. Also, intestinal, they can just live in the water. So this is a little bit different, is that cestodes don't just swim in the water. Flukes do, or trematodes do. So they do actually just swim in the water. So a person can get it just from the organism living in the water itself. But also it can be foodborne, so it does burrow into the tissue and then the person can eat it. Also, it can be, why did I say tissue, waterborne? Because it can burrow straight from the water. So a person that works in the water, like a person who fishes a lot, who stands in water, a person who washes their clothes in water, a person who hikes and stands in the water, a person who, anything like that you might think of how they might have contracted it, but they can swallow the water and it can be intestinal, or they can be standing in the water and it can dig into their tissues from standing water. So I don't know if you can see this. This is probably the most disgusting thing you've ever seen. Top 3 is that this is the person's, it looks like their trachea. And then this is a fluke right here on the lining of their trachea. Okay. So snails are their primary intermediate vector. They do have other vectors as well. So sometimes people can get it from their pets, from wild animals, birds, insects, things like that, shellfish. Okay, so let's go into roundworms. So roundworms, we'll start talking about Ascaris. And this is going to be one of our no cards, so it'll be easier to remember. But Ascaris looks like a traditional worm. If you see something that looks like a traditional worm, that's what it looks like, except they're yellow. They have the same life cycles. So the larvae are able to burrow. So the larvae can be anywhere, lungs, liver, intestines. They can cause little granulomas. But the adult traditionally likes to live in the intestines. Transmission is fecal-oral through the ova, through the eggs, but also it can be foodborne because the larvae can burrow. Everybody grossed out? Okay. So yes, these are the ones that look more like traditional worms. So a patient, so one thing that this Ascaris likes to do is the larvae, what it can do is it can burrow into the lungs, and then the patient can cough up eggs and swallow them, and then the adult will live in the intestines. So coughing actually is one of the symptoms of a helminth infection or helminthiasis, is because some that can dig into the lungs will cause the patient to cough and then swallow, and then it lives in the intestines because the larvae will live in the lungs or some other area. Fecal oral foodborne, do I want to say one more thing about that? No. Okay. So here are pinworms. So pinworms do also look like worms, but they are tiny, tiny, tiny. So pinworms are actually very common, probably the most common out of all the ones we're talking about. Ascaris are actually really common too. I have a statistic on that. Like, you really don't want to hear it, but I'll have to tell it to you. So 2 billion worldwide. How many people are there on the planet? 8 billion? 2 billion people have pinworms. It's actually really common in the United States. 20 to 40 million people. That's a lot. That seems way a lot. But also it's a lot of children. This will pass easily between children. And that's why they, like, sandboxes have kind of gone out of favor, is because animals will defecate in a sandbox and then the kid will play in there, and the kid can get pinworms that way. 50 to 100% of institutionalized persons, I just read that, like, that seems crazy and really sad and terrible. And my guess was, I was like, why would that be? Like, why would it be institutionalized persons? And my guess is because of food standards. It's just my best guess. And then just being able to control transmission in areas. So fecal oral, and then what they'll do, this is gonna be probably the most disgusting thing you ever heard in your life. This might make number one, is that the pinworms crawl to the anus and they'll lay eggs anally. So they lay eggs extra anally, like around the anus, and then crawl back up. And so when they're crawling to the anus, that children, it's very itchy. So children that have like itchy bum, they're like itching, and then it's because the pinworm will crawl down and lay eggs perianally, it's called. Disgusting. So what they do, what you'll hopefully never do, but you might have to do, is called a tape test to see if your patient has a pinworm infection. So for children, they'll have like crankiness, they won't be able to sleep, they'll be itching their bum a lot, they might just be generally irritable and not feeling good or not eating, and so it might tune you into that it might be a pinworm infection. So Enterobius formicularis is one of our note cards as well, and I'll tell you more about what the tape test is and that unfortunate time later on. Okay, so those are pinworms. And then whipworms. So here, these are also fairly common. And these ones look like the Indiana Jones, like, they look like a little whip with a handle. I probably didn't draw it very well, but they have a thicker side and then a thinner side. And so they are whipworms, intestines, fecal oral. So these are primarily fecal oral and not burrowing. But what they do is they'll attach to the intestinal wall so they can cause dysentery because it's breaking down the intestinal wall as they're living in the intestinal wall. So I didn't want you guys to remember any of this, but I just put it on there, is that dysentery will be more common with whipworms. Something called rectal prolapse. So it's almost like hemorrhoids, like when the rectum comes out of the anus, and it's generally because of the wall of the colon is not intact anymore. So it's not holding in the integrity of the colon is damaged because of these helminths living within the tissue. So it can come out of the anus, which I can't imagine is very pleasant. Long, slow growth, so somebody can have these for a very long time. Okay. And then our last one are hookworms, and these ones do look a little bit more like traditional worms as well, except they have one little tiny end. So they look like a traditional worm, and then they have this tiny little tail. And hookworms have like teeth. So these hookworms, oh gosh, I'm so excited to tell you this terrible story. So when I was in my teens, I was probably 19 or something like that. I went to Fiji, and I lived in Fiji for a little while, and so everybody that would like travel around the day, and then we'd come at night and everybody would drink beer and sit around the table and laugh and joke, and from all over the world, you know, people meeting each other. And I met this guy, he had been working for the Peace Corps in a place called Kiribati, or it's called Kiribati is the name, how they say it themselves. Have you heard of that island before? Yeah, so there's like millions of people living on this tiny, tiny island, and so they have all kinds of problems with like plumbing and with housing and food shortages and all this kind of stuff. So they have this huge overpopulation. And so we're sitting there and drinking, and he's like telling these stories, and I was like super into his stories, and I just like looked down for a second, and I was like, you know, he's talking and my mind is like screaming because they're crawling, like he had worms crawling under his skin. And so I was like, oh my gosh, be calm, be calm. And so I asked him, and I was like, what is going on with your arm? And he said, oh yeah, I have a hookworm, it's been in there for a while. I don't want to get it out until I go home because infection is so rampant. And they said it's fine, so I'm just gonna leave it. I was like, oh, don't leave it. I would be like, next plane home. I go, bye-bye. So they just can live in the subcutaneous tissue, and they'll just be stuck in that dermal layer, and they can just live there for a long time. So hookworms are incredibly, like, the most burrowing of all of the helminths, and but they also can live in water, in open water. So I don't know how he got it in his arm, maybe from washing or from something like that, but it got into his arm, and but it can get into people's feet and things like that. And sometimes it will just die there, but also sometimes it can cause all kinds of issues. Sorry, go ahead, Dr.
What is happening in the bottom picture? Is it burrowing out? Can it do that?
It can it burrow out? That is a really good
Or is
question.
that what's it depicting in the picture?
Yeah, it's depicting it burrowing in.
So
Yeah, I wouldn't think it would burrow out because it's not the area. It's like you know, they might have like. Yes, that's what it wants. Warm, and it wants food.
how does it get in?
Because it has like literal like teeth. It has like
wouldn't that be painful to the person?
you know, I do think it has like numbing. It doesn't. The person doesn't feel it. Yeah, but that is a good question. I was curious about that as well. Yeah.
You said these were the most burrowing?
Yes, the most. So larvae do burrow as well of the different burrowing ones. Anything that is foodborne can burrow, whether it's the larvae or the adult. These ones, this is their primary method of transmission, even though they have other methods of transmission. Sorry, go ahead.
That was actually my first question, but I was curious also, are whipworms the only one then that won't burrow?
That is a good question. I think I have a thing on that. Transmission, fecal, oral, foodborne, fecal, oral. Oh, pinworms as well. Yes. So pinworms and whipworms are the only two that do not burrow. The rest do. Great question. Okay, so here we are with hookworms, and there are many different kinds of nematodes that are infectious. They're from all different branches of life, wherever they come from. And yeah, I just wanted you to know there's a lot more, and I covered the most common ones. Yeah?
Do we need to know these ones too, or is it
No.
just out there?
No, and the only ones that you need to know their genus and species are the two note cards, and that's it. Except Ascaris, because that's they're just called Ascaris. Pinworms, whipworms, hookworms. That's a genus name as well, but they're called that. Okay. So here's our little summary. Repro, I don't care that you know that. I do care that you know transmission. So this is, I just put these on here, just the most common methods that how transmission happens. So it should all make sense. If you put fecal oral, you know it's the 5 methods of fecal oral transmission. If you say foodborne, it's burrowing. If you say waterborne, it can free float in the water and get in that way. So you don't have to memorize any of these. I just wanted you to know those and read those. And then some characteristics about these, and I do want you to know these because these are things that are like identifiers about those specific helminths. Okay, so that is it. So we're going to do our note card, and so what we're going to do today, did we do Trichomonas vaginalis? What did we do last time? What did we do? We did Giardia. Okay, so we're still on protozoa. I think this is our last protozoa. It might be. I'm not sure if I'm going to do toxo last time, but okay. So here's Trichomonas vaginalis. This is our last protozoa. So what I'm going to do today, actually, after we do our note card, I'm going to give you a little 411 about the unknown project and kind of what our next steps are and how we're doing with that and how we're feeling about it. And then what I want you to do is sit for a few minutes after we talk about it, look at your packet while you're up here. I'm going to head down to the lab. Look at it while you're up here and just kind of start making a plan if you haven't already. If you already, like, I already spent an hour on it at home, I feel good, I'm confident, I know what I'm doing. If you haven't spent any time on it, I want you to stay up here for a few more minutes, 10 minutes or something like that, and just like, okay, what is my plan for the day? And then I'll just give you a little bit more information about it. Okay, so here's Trichomonas vaginalis. I wanted you guys to be able to get out of the lab early today because I want you to be able to go and vote if you haven't voted yet. I already went this morning and it was pretty busy. And if you guys get out of here at 4:30 and try and go vote at 5, you might have to be waiting in line for a long time. So if you haven't voted yet, or if you can't vote or you're not going to vote, no judgment, it's up to you. But if you do want the opportunity to go and vote, I want you to be able to get out of here a little bit early just so that you don't have to wait in forever long lines. So I want to try and get done with lab a little bit early if we can. I already voted this morning, so if you haven't had the opportunity to, it might be a good idea not to leave at 4:30 and be at the ballot box at 5 o'clock and be like, oh my gosh, I have like an hour and a half line to stand in. They've been moving people through. There was a line when I got there this morning, but it took me like, I don't know, 12 minutes to get through the line, so it wasn't that bad at all, but you never know. It depends on the place that you go. So the reason I wanted to bring up this note card is because now we have some comparisons. So I'll show you some pictures. 70% no symptoms. It's frightening, right? If somebody has an infection, you want them to have symptoms, right? So that they're not transmitting it, but complications during pregnancy, as you might imagine. 3 million people in the United States, 200 million worldwide. Okay, so let's do some comparisons really quick and then I can go back to that one. So here is our bacterial infection. So this is bacterial vaginosis, or BV. So make sure to compare these and that you understand the difference between these. So I don't want to call them STIs, but we can call them vaginal, or sometimes it's penile, but not always, infections. Primarily vaginal, and unfortunately, females are, I don't know, there's a giant quantity more likely to be able to go and get treated where men are not. But also, women are more sensitive in that way because of the urethra is separate than the vagina. So in males, because of urination, sometimes they won't have so many symptoms because it's like flushing out, and then even though they still do have an infection. But for females, one option is BV, or bacterial vaginosis. You know this. From Gardnerella vaginalis. And that is not an STI. That is from a increase in pH and yeah, I guess I can put that over here. Increase in vaginal pH. Okay, so then our other one that we talked about is a yeast infection. And that is from Candida or candidiasis. We'll call it candidiasis. So bacterial vaginosis, candidiasis from Candida albicans. And this is not an STI. This is from change in pH also. Then we had, but also don't forget we have our other STIs like chlamydia and gonorrhea, things like that. So don't forget the other STIs. But one more I'll mention is TB or trichomonas. We wouldn't call it trichomoniasis. Yeah, we would. And this is Trichomonas vaginalis. And this is an STI. Do you want to put the other ones up here? You'll have to help me spell them though. Here's bacteria. Here's fungi. This is protozoa. So we have chlamydia. How do you spell it? I'd have to pull it up. Nope. Let me pull up the note cards. Trichomonas. That's right. And then our other one was gonorrhea. Oh boy, you have to help me spell with the spelling. Gonorrhea. Oh, it's. Is that right?
Yes. No, R-A, no I. R-A. And then Chioeno, R-R-H-O.
Whew, that's a tough one. Okay. So this is chlamydia. It's called the infection. And this is called gonorrhea. And this is bacterial. And this is bacterial. Okay, so those are all of our infections, yeah? Does that encompass everything? So here, if it's VV and you do a vaginal or a penile swab, you'll see, well, not penile in this one because it's not sexually transmitted, but this is an erythrocyte. This is a skin cell, and here, around here at 400 times, it looks like a clue cell. So these are the clue cells that give us a clue that your patient has VV. They have to be treated very differently from other types of infections. So here's candidiasis. Remember what this looks like? Spaghetti and meatballs. Here are the fruiting, well, here's the yeast. And here are, it looks like pseudohyphae. 400 times, that looks very different than here, right? And then trichomonas, they're large. Here's an erythrocyte, here's a trichomonas infection. Okay. Okay, so what I'm going to talk to you about, let's talk about our unknown project. So far, if you've been planning and thinking about it, which would be a great thing, we took our original number and we plated it and did a 3-way street plate. So hopefully we had 2 colonies. So this is your goal. So if you did not get this today, or you do not think you have 2 colonies, or you're worried you might not have 2 colonies, this is priority number 1. Make sure you have 2 separate organisms. If you're not sure, it's a good idea today, if you haven't done it already, to plate onto a PEA and a CV. So this is one option that you can do. So this is phenol ethyl alcohol. And this is crystal violet. And this is selective 4 gram negatives. This is selective 4 gram positives. So this is a good option, is to see, okay, then what you can do on these PEA and CV plates is to match to the original colony. So here you had colony A, you had colony A, and you had colony B. So looking at your CV and PEA plates, is one all colony A and one all colony B? Or is one blank and you have 2 here? So you can start comparing, which one's A, which one's B? Do I have a gram positive, do I have a gram negative? Do I have 2 gram positives, 2 gram negatives? What do I have? Another option that you can do now is do a gram stain. So I'm gonna be looking at my gram stain, and I can use this is your choice. So think about it in the next few minutes when you're here by yourself, what would be the most time efficient? Is it most time efficient to do these separately and see if I have a gram positive, gram negative? Is it most effective to do them if you're not 100% sure, gram stain that. Do you have positives and negatives in your gram stain? One problem that many of you are gonna have that I wanna make sure that you're clear on, sorry, Scott. So you have, hopefully you also did a slant. And you have an A and B. Hopefully, if you didn't, it's okay, you can do it today. So you want to keep that backstock fresh every time. You always come in, make an A and a B backstock. Date it, get rid of your old ones. Except your old ones you have to use today. You have to make the slants, but you have to use it. Use the stuff to start planning your method of attack. So what you can't do is you can't use this crystal violet and gram stain it, because that crystal violet is going to be purple, and the purple won't go away. So you have to use either these, or this. Yeah.
Can you take from P and CV and put it onto slants to grow more?
Yes, you can, and eventually the CV will grow out of it. So if that's your only options, if like I have my CV, I want to use that, it will eventually, as they replicate, the CV won't replicate with it. The CV will grow out of it. But you can't gram stain it today. You could probably gram stain it on Thursday.
Like for me, I don't know if anyone else too, it was really hard for me to, recognize the 2 different organisms on my isolated string plate.
Mm-hmm.
So it would be okay not to do the gram stain today, but to grow more and do that the next.
Yes, but
Okay.
I wouldn't wait. I wouldn't just do this and then be done for the day, because that's your whole day. So I would do something else other than planing, and then you could do your gram stain at any point. You could do your gram stain later, but do some other tests just so you use your time efficiently. I'm sorry, really quick.
What do you mean by grow out of it?
So it will, the first day, so if you take, let me switch these colors around really quick. So your PEA is just gonna be yellow, like the normal bacteria. CV is going to be crystal violet color. It's going to be like a purple. So if you put this on your slant, it'll be purple today. And even on Thursday, if you take this and inoculate a new A and a new B, this might still on Thursday be just like light crystal violet color. So it might still be hard to gram stain that.
Okay.
But eventually you will be able to gram stain that, because some of these that were living in here with crystal violet are still living here, and some are still living here. But every time they go through binary fission, they don't have crystal violet in them anymore.
So what would you recommend using to inoculate the PEA and the CV? Plates?
I would recommend using your originals.
Okay. So we gave the originals back to you, right?
Yes,
Okay.
you have all your originals. Yeah. But you can use anything, really. I mean, you can try and see if you can get, I don't know, that might be tricky.
Okay.
But we can work together on that one if you have a
Okay.
question. Did you have a, sorry, go ahead.
I'm just trying to, in general, clarify the whole thing, like backtrack a little bit. So it's like we have the plate. Ideally, there's the two different organisms on it that we are able to physically identify. So when we come into the lab, today especially, but in general, what we want is to take a media that has both of them on it, make two new backstocks by separating them into like new slant tubes. Like in this case, we would be taking the petri dish from last time and then making an A from one organism and a B from another organism, new, like fresh. And then we would be using each of those today, ideally, for like the gram selective tests, but not doing a gram stain until next time.
Yes, you can do that. So did you make slants last time?
I did.
You did. Okay, so if you made slants, then you should be on your way, because you can just use these to do tests now. But to make sure that these are either gram positive or gram negative, you can take these and gram stain these today and check. But also what you can do is plate these on, you can take these if you're not 100% certain, and you can do like this. Does that make sense? Like a PEA and a CV. So you plated, you took your A, you took a little bit from your A, wiggled on a PA, took a little bit from your B, just label them clearly, a little bit from your A, a little bit from your B, and confirm. Does that help?
And so when we're down the road, like we're past this step, let's say we go in for level 2 or 3 or whatever, when you say make new backstocks, we take the 2 slant tubes that we had from last time and then we inoculate them onto new slant tubes.
Yes, so you'll be taking slants to slants every day. Every day that you're in the lab. Yes, yes.
And is
because
that,
you can kind of get rid of these now. I mean, as long as you checked this with us, you did all that, then you can kind of get rid of all that. And then I would take pictures of it just in case. But here's what you're going to be doing all the tests on every week.
and that's to keep them like fresh, so to speak.
And that's just to keep them fresh.
Okay.
Yes, yes. Awesome. Great.
What happens if you don't continue to do that? Like, do they
So
die?
between
Or?
they'll just die, yeah.
Okay.
Yeah. So between a Thursday and a Tuesday is longer. Between Tuesday and Thursday would probably be fine. And then also if you put it in the fridge, they'll be better, because remember that bacteriostatic is like then they won't divide as rapidly and they won't need as much food. So fridge is always a good option too. But we always have to be careful with our space utilization, because we can't keep everything in the fridge. They don't like us to, because they have to use that fridge for other things. So yeah.
You don't have 2 definitive colonies for science, or you have to have 2 definitive colonies to do it. Like for example, if I did an isolation streak Tuesday, and at this point it doesn't come through as 2 separate, my original might be too old.
So but you did this too, right? You did the PEA and CV.
Yeah.
So what I would do is if you don't have 2 separate, if this didn't work, now use these and make these. Make your slants, one from a PEA, one from a CV, if you have a gram positive or gram negative. And then from there you can continue your backstock. You just won't be able to gram stain that. And then what you can do from there is you can separate those out to a PEA and those out to a CV to see, okay, do I have 2 gram positives or do I have 2 gram negatives or 1 gram positive, 1 gram negative?
And you can still do that if it's like 2 inside the PEA as opposed to 0 in the CV. They're not separated there either.
Yes.
And just work on separating it a different way, just, fresh stocks, so I understand.
Yeah, if that's the case, let me know and I can help you with that, yeah. Yeah. Oh, yes, sorry. Okay, so as you can tell, it gets complicated. Just stay clear on your steps that you're gonna take. You can start running tests. There's everything down there. So there's going to be every test you could ever possibly run down there. So you can start running tests. Just take a minute, take a breath before you run a test and think through it. Okay, if this is what I think it is, I'm just gonna start with this test, and this is a good starting place, and then I'm gonna run these tests successively. So don't try to plan it out a little bit. Yeah?
Are we starting with wrapping up block 24, 25?
Yes, we do have to finish 24, 25, yes. Yes.
Oh, we're starting with that?
Yes, we're gonna start with that,
Okay.
yeah.
So if we run a test and narrow it down too fast and we don't get our 4, is it okay to go back and
Yes.
insert
just
a
run
test in
another.
between?
Yes, you can. You can run other tests in between. If you already figured it out, say what test would have been useful
Okay.
if I'm gonna show it to another scientist and they're gonna run it. What
Okay.
test would I, if they didn't pick this one that was my definitive, what could I have, how could I have led them there with 4 confirmatory tests? Because
Okay.
also your patient has gonorrhea and you don't wanna be like, well, I did these tests. And you're like, well, are you sure? Like, we want 4 confirmatory tests,
Uh-huh.
yeah.
Thanks.
Sorry, Dr. Villegas, did you have a question? Did you have a question? Okay, okay. So if you guys need time to plan,